So I think that we are ready to move on and introduce our speaker. So Elliot, if you wouldn't mind. Hi, Elliot, how are you?
I'm great, Laura. How you doing?
Good. I'm so glad you're here with us today.
Yeah, me too.
Um, so you have been collaborating. Well, let me introduce you first. So Elliot holds a PhD in ecology, evolution, and systematics. And until very recently, he was an employee of the Cornell Lab of Ornithology. But he recently joined the American Bird Conservancy to work on automated acoustic monitoring and conservation projects. So congratulations on that move.
Thank you.
Yeah. Elliot retains a deep interest in avian phylogeny and continues to lead a project to build and update a layer for a phylogeny layer for birds of the world. So we're super excited about this work, and we'd love to hear more from you. Um, so I'd love you to kind of, today you're going to explain that vision for aligning, for creating a phylogenetic layer to birds of the world that adapts with taxonomic revisions. So that will be very important for many reasons. And I hope that you can explain that all to us in the next hour or so. So the other thing that Ellie's going to do occasionally is drop a poll in our right in front of you. So we're going to have a little bit of interactivity today, which is kind of a new thing for us, but that should be fun. And yeah, so if you're ready to begin, we are.
Okay,
Okay.
cool. We just crested 650 participants.
That's a lot.
I'm excited.
That's a lot.
All right, share screen, share sound, desktop 2. Are we in there? It's looking good.
Yes, it looks great.
All right, awesome. So yeah, as Laura just said, I'm Elliot Miller from the American Bird Conservancy, and I'm really excited to share with you what we've been working on now for the last 4 years. All right, so this is a collaboration with the Open Tree of Life project, which is an NSF-funded project to create a dynamic and updatable phylogeny for all organisms in the world. I want to start by acknowledging my close collaborator, Emily J. McTavish on the Open Tree side. This project, the Open Tree of Life, is really ambitious. It's super successful, and they have lots of awesome tools, both in terms of their back-end software and in terms of their front-end visualization. And it's really terrific for building big trees across lots of taxa. But one thing that we have found is that in order to bring the resulting phylogeny up to the quality we expect for a taxon as well known as birds, we need to do a lot of sort of careful hands-on curating and data management. So in other words, we need to take those awesome tools that Open Tree of Life has built, and we need to push them further in new ways. So much of what I'm going to talk about today is simply going to be showing you what that means. But this is a really big moment for me, and I want to just dwell on it for a second. All the awesome existing Open Tree of Life software notwithstanding, for me, this project's always been a bit of a late night, weekend, while watching TV in the basement kind of thing. So for 4 years, we've been plugging away at this project to bring it up to where it is today. And I can't help but be reminded of a commercial I saw when I was younger, and somehow I managed to find it and dig it up. So I hope you'll humor me.
My apartment has a TV. I watch RPM tonight for my knowledge. The more I watch, the bigger it gets. Someday, I will share my knowledge with the world. That will be a fun day.
Oops, nope, there we go. So today is the day, folks. I want to start by making sure that everyone is on the same page with jargon. I promise there'll be some fun stuff soon, but I want to just get a few definitions across the line and make sure we're all on the same page. So the specific terms I want to get across today are taxonomy, phylogeny, and clade. And there'll probably be some other ones that I accidentally mix in. So Laura, cut me off if I'm, saying something that's confusing. Just interrupt me and I'll define it.
Okay, we'll do.
So taxonomy is the hierarchical classification of species, where every species, when it is described, gets what's called a binomial, a genus-species combination. So for example, take Heliomaster longirostris, the long-billed sunseeker. Remember, scientific names are in Latin, so you've got the adjective after the noun, like Spanish or Portuguese. And these are what you'll often hear referred to as a species scientific name. So these are organized into genera. Species are grouped into genera. Genus is a single one. Genera is the plural. Heliomaster in this case is the genus, right? Genera are then organized into families, families into order, and orders into classes. This is a hierarchical classification system, nested categories. It's not a hierarchy in terms of one taxon being more derived or better than another. It means nested categories. All right. That's not to say that some species can't be so weird that they get their own order. For example, the Watson gets its own genus, its own family, and its own order. It's just that weird. So originally, when Linnaeus first developed the classification system that we use today, Darwin wasn't even born. But it turns out that the concept of a hierarchical classification system lends itself really well to the concept of evolution. So that's a lot of words. I wanted to break it up again with a short quiz. And let's start with the familiar sort of downy woodpecker, for our North American audiences at least. I'm going to show you these three other species here. And the question is, which of these do you think is the closest relative to the downy woodpecker? So Laura, you can go ahead and roll that poll. I gotta see if I can watch it come in.
Okay. Let's see.
Yeah.
All right. Everybody see the poll? Oh, look at that. A lot of people think it's the hairy. Let's see what we end up with.
Yeah, maybe give it another 30 seconds or so.
Okay.
See the results coming in. So I'll need you to summarize them for me.
Mm-hmm. All right, I think we'll end the poll there. So we have 54% believe that it's hairy woodpecker, 16% believe it's white-headed woodpecker, and 30% lesser spotted woodpecker.
Cool. All right.
How we doing?
Here, I got to get my Zoom thing functioning right. All right. It turns out it is the lesser spotted woodpecker. Many people probably chose hairy woodpecker as their closest relative. And for years, it was assumed that this was true. They look really similar. You'll often find them in the same forest together, and they were traditionally placed in the same genus. Now, however, we know this is the result of convergence. And in fact, downy is more closely related to this lesser spotted woodpecker of Eurasia. Cool. Time for another quiz. This one's for Kevin Burns if you're in the audience. All right, so let's start with Western tanager. Which of these options do you think is the closest relative? Could you roll that second one? Thank you, Laura.
Sure. So the options are northern cardinal, Brazilian tanager, or red-crested cardinal. And here we go. With 700 people, we do have to wait a bit, don't we?
Yeah.
You are popular.
Birds are popular.
Okay, just about 500 people have entered.
Cool. And how are things looking?
They are looking as if Brazilian tanager is the most closely related.
Yep.
All right.
Give it 10 more seconds.
Okay. All right, we're going to end poll there. We have, and the results show 51% for Brazilian tanager, 27% for red-crested cardinal, and 23% for northern cardinal.
All right. Yeah, this is another trick question for you. Many folks presumably selected Brazilian tanager because it's got the same name, tanager, in it. But actually what we now know is that we might as well start calling these things Western parangas. So Western scarlet and summer tanagers are in fact cardinals of the family Cardinalidae. That means they're more closely related to northern cardinal than they are to any of the true tanagers, like Brazilian tanager. And actually, I threw that red-crested cardinal in to really throw you off. That species, native to South America, but actually introduced and widespread in Hawaii as well, is actually a tanager also. So a lot of fun stuff going on with convergence in that family. And now I'm going to get back into some definitions. So if I go like this, can you all see my cursor? Hopefully.
Yes.
Great. Okay. So this is, well, it froze on me now. But anyhow, yeah, here we go. This is Darwin's first tree, and it's a good introduction to the idea of a phylogeny. So the way to read a phylogeny is as follows. First, you've got these species out here at the tips. These are extant, meaning that they're alive today. And then as you work your way from right to left, you're going back in time into the fossil record. When you hit junction points like this, these junction points represent shared common ancestors. So this, for example, is the common ancestor the last time that species A and B shared a common ancestor. And the farther to the left you go here, the more distantly these species, these lineages shared a common ancestor. So this, for example, is when species A through E shared a common ancestor. All right, so that's a phylogeny. If I was teaching a class, I'd say any questions, but I'm just going to move on. So you can easily map taxonomy onto a phylogeny like this, where, for example, this node, this common ancestor, represents the progenitor of all the species in the genus Picoides. And I'm putting that in quotation marks here. And that's because in this figure, Picoides does form a single group on the tree. But as you'll soon see, that's not quite right. And then here, for example, we can show where the common ancestor of all Vanilliornis species lies. So we can map taxonomy to phylogeny, usually. And this brings up a critical point, though. When the Linnaean classification system first became popular, and for centuries afterwards, people weren't necessarily concerned with whether all the species in a genus or a family or whatever actually shared a common ancestor. Taxonomists were just grouping species based on gross morphological similarity. And then over the centuries, scientists began examining species in greater and greater detail, striving to make those groupings reflect true similarity and not derived similarities. But morphology can only get you so far. I'm sorry if I offended anyone there. But for the most part, it's great at helping you group species into families and such. But relationships within those families can be really hard to resolve. And it's hard to preserve and stare at the syrinxes of every one of the 11,000 species of bird in the world. So another thing to point out is that there are many more nodes in a phylogeny than there are named taxonomic levels. And this means that all else being equal, phylogenies can tell us a lot more about how things are related to each other than can taxonomy. Fortunately, scientists figured out how to use genetic information to reconstruct phylogenies. And in the process, taxonomists have become increasingly concerned with ensuring that families, genera, et cetera, can all trace their roots back to a common ancestor, to the exclusion of other species. I'm going to show you what I mean here. So I'm going to introduce you now to the last term of the day, the idea of a clade. Look at this phylogeny. A clade is any branch and all its associated sub-branches, twigs and leaves. So twigs and leaves from other branches are not part of the same clade. So here's what I mean by a single branch. That's a clade. Things from over here are not part of that branch. They're not part of that clade. Here's another clade as well. Notice that a clade doesn't have to be a named taxonomic entity, but it can be. So here, these Picoides species form a clade, and then these three random Vanilliornis species form a clade as well, an unnamed one as far as I know. All right. Ideally, all named taxonomic levels are true clades, like I just said. But the more we use genetics to study these things, the more we find out that some of these genera and families need to get redefined. Here, once we zoom out a bit, we see that some of the rest of what were once called Picoides woodpeckers are in fact part of a separate clade. So these things up here versus these Picoides, this is not a true clade, right? Like these belong to different branches. Just a little note, American and Eurasian three-toed and black-backed woodpeckers are still Picoides. They're somewhere totally different in the woodpecker tree. So we do still have Picoides, but all this stuff gets renamed now, and I'm going to show you in what way. So we're going to dig into that downy hairy example from the first quiz. These two species were once in the same genus. Here's downy, here's hairy, lesser spotted. But when we found out what the true relationships among these species looked like, we had to do a little bit of renaming. So, you could do that in a number of different ways. The IOC, which is a different taxonomy than used by Birds of the World or eBird, they took the fine-toothed comb refined approach. I put the fiat up there. So like some of this species here, in Dryobates, all the lesser spotted downy, those have been moved to the genus Dryobates in a restricted sense. The hairy, smoky brown, white-headed, red-cockaded, all that stuff gets moved to Leuconodopicus, and then Vanilliornis, I think at least, still exists in the IOC approach. The Clements takes a different approach and just lumps them all into Dryobates. I mean, they are woodpeckers, right? And neither of these is more or less scientifically correct. They have the end result, which is making sure that taxonomy aligns with phylogeny. All right, that stuff's pretty in the weeds. I want to move on to another fun quiz.
Elliot, before you do, somebody did ask, what do you mean by convergence? And I thought maybe we could answer that before we go much further.
Yeah, this quiz will be a great example of that. Oops, maybe I gave away the answer. Convergence is the phenomenon by which species that share similar ecological characteristics, a similar niche in the ecosystem, have evolved to share similar characteristics, and that the fact that they share similar characteristics is something that has evolved over time as opposed to being inherited from a common ancestor. Cool. Yeah, hopefully that answers that. Something you could talk about at length, obviously. So here we're gonna ask who the closest relative to the tawny-crowned honeyeater is. Your options are Cape sugarbird, gibber chat, and magnificent sunbird. How are things looking, Laura? Laura, you might be on mute, so let me know how the results are looking.
Oh, sorry, I was on mute. That's my thing. I'm always on mute.
No worries. Yeah, how do the results look?
Well, this is amazing. We have a three-way tie. Cape Sugar, Magnificent Sunbird, and Gibber Chat are basically all 33-34.
Yeah, that's really cool. And I guess I might have given too many hints there. But this is a really neat one. They only started getting a hint at the answer to this one in the mid 70s, I think, when they started looking at the tons of these chats and realizing that they looked an awful lot like honey eaters. So sorry, get the results up there for you. Yeah, a gibber chat is not a true chat at all, but it's actually just another honey eater species. The other two things, the nectarivores, the sunbird and the sugarbird, just look like that because they also go to flowers, and that's a really useful morphology to have for flower feeding. So that's kind of a nice example of the convergence you were talking about, somebody was asking about.
Thank you.
All right. So now let's dig into the actual project and take a look at what we've been doing and why. So from the 10,000-foot perspective, we're making a global avian phylogeny that has one tip in it, no more, no less, for every species in the Clements taxonomy. The taxonomy used by Birds of the World and eBird. This product is updatable, meaning it's easy to add new information to, and it's dynamic, meaning it's possible to remap tips in taxonomy and get slightly different outputs. All of this is tracked and versioned, of course, and critically, what we're doing also tracks the source of the information itself. And I'm going to show you why that's critical in a moment. To dig into the why of why we're doing this, for starters, the process of phylogeny publication and taxonomy revision are usually two separate steps right now. Findings are published all the time that challenge current taxonomy, although fewer and fewer these days. Thank you very much for all the hard work, taxonomists. But at any rate, we don't really have a centralized place where taxonomists can look to see what might need renaming. Taxonomists need to watch the literature, gauge how good the support is for renaming, and then make a decision about how to proceed. And so what we're doing here, as I'll show you, can hopefully help streamline that process. Another good reason to do this project is because phylogenies are actually necessary ingredients of modern scientific analyses. I'm going to give you a brief and grossly oversimplified primer on phylogenetic comparative methods. So the reason phylogenies are necessary here is because closely related species are expected to share traits. If we want to know, for example, whether blue coloration has evolved in birds that eat fruit and red coloration has evolved in birds that feed on flowers. Oops, I didn't mean to go ahead. We need to statistically disentangle what is expected based purely on evolutionary relationships. So for example, if all blue birds are part of the same clade and all red birds are part of the same clade, it's really difficult to say whether fruit-eating birds have evolved to be blue or whether this is just a coincidence. But if we have this array of traits across the phylogeny, we have a lot more power now to make the statement that fruit eating leads to blue coloration in birds. We have this multiple origins and shifts in state that are correlated with shifts in diet. And now we have more power to make these statements. So scientists need phylogenies like this to run analyses and disentangle these factors. To date, scientists have either sort of restricted themselves to phylogenies that they custom make for their research, or they take advantage of previously published phylogenies and then prune them down to the species that they want to focus on. So the most famous of these phylogenies that scientists use, even today, is one that was published in 2012 by Walter Yates, Gavin Thomas, and others. That phylogeny has been used now 3,287 times and counting. It's been a game changer, but it's also old, both in terms of the relationships it shows. I mean, it pretty much predates the whole genomic revolution. And in terms of the taxonomy it uses, there are only 9,993 species in that tree, which was nominally all the species in the world at that time. But actually there's 11,017 recognized in eBird today. There'll be more next year. And so the Yates tree has been awesome and it's facilitated a ton of great science, but it's time for an update. And ideally one that will be dynamic and grow and evolve over time. Another good reason to do this project is that the taxonomic crosswalks we're building can help align large trait databases like the AVONET morphological database for all birds globally. It can allow us to impute missing data when taxonomy changes, and it allows us to keep these databases aligned and current going forward. And the last reason we need a project like this is because we can use it to teach people about evolutionary concepts. Birdwatching and listing, all this stuff is totally powered by evolution, but a lot of people don't realize this or think about it. And there's some great opportunities like this webinar to sneak a little bit of evolution into the mix. All right. So, I want to take a minute to say, to make this global phylogeny, we're really standing on the shoulders of giants. Science always has progressed by building on what came before. But this project really takes it to a new level. A key goal of ours is making sure that contributors, including the museums and curators that power a lot of this work, get the credit they deserve in helping build the final product. I'm going to show you how we do that. And I just wanted to also point out that OpenTree works by incorporating published phylogenies. That means we need the actual tree files themselves that generate a phylogeny like this. And we don't actually work with raw genetic data for this project. Someday, yes, maybe, but for now, we need these tree files. So authors, please archive your actual tree files when you publish papers. It will also help make your work more reproducible. So as I said, we're taking these phylogenies and we're curating them, entering them into the Open Tree of Life system. That means I needed to go through the literature and find any published phylogeny I could get my hands on. That applies both across families and within families. At this point, I've carefully gone through all of the 250 or so bird families and found the best available phylogeny for each, and then I've entered it into the system. And we also have family level phylogenies like this recent Stiller one in Nature that I know some folks were interested in. that have been entered as well. And so more amazing trees are being published every week, and we're adding these as they come out. And it's also worth reminding people that this is really a community-led project. So like, I definitely curated quite a few trees, but there were already tons in the system from people who came before me. So a big thank you to everyone who did that. Okay, so we're assembling these tree files and we're entering them into the Open Tree of Life system. I probably should have grabbed some screenshots to show you what that looks like. Go check out the Open Tree of Life curator system if you're curious. When we do that, the paper DOI gets linked to the phylogenetic information, and then we have to carefully match tips from the trees to taxa in the Open Tree taxonomy. These trees then get stitched together to build a composite tree linking all the species in each tree together. In total, at this point, we've entered 278 phylogenies, about 41,000 tips we had to match, and that gives us data for 9,233 species of birds. Remember, we're talking about just birds here. Open Tree of Life actually deals with all life, so their database is much larger than just those 278 trees. Another thing to know for those who are interested, when phylogenies conflict with one another, which they do, we defer to the most recently published phylogeny as the sort of correct answer. And that allows things to evolve and improve with time, ideally. So hopefully by this point, you're getting the sense that as new trees come out, it's easy for us to just add them and improve the process. All right. To help drive the point home of how this super tree stitching process works, I'm going to show you this super slick animation I made with graphics circa 1993. It might just serve to make more questions, but I'm going to give it a go and hope it helps. So in 2019, Oliveros and colleagues published a great study in PNAS that puts forward a phylogeny for most of the passerine families in the world. And I'm showing a portion of that here. And then within the dashed lines, I'm drawing attention to the suboceans, which are the flycatchers and furnariids, antbirds, antpittas, all that stuff, suboceans. So they did a great job of showing us where in the tree of life suboceans plug in, but their study only included about 20 species of suboceans. It is actually a very large clade of birds, largely restricted to the New World. In 2020, Mike Harvey and colleagues published a paper focusing just on suboceans. This was an awesome tree that covered 1,287 of the 1,306 described species of suboceans. There are probably a few splits that are needed. And so using some fancy magic PowerPoint animation, we can just stitch the Harvey tree into the Oliveros tree, and now we're off and running. This is obviously a huge oversimplification of what's actually happening under the hood, but I'm hoping it helps bring people along that might otherwise be unfamiliar with these ideas. All right, after we step through all these tree addition and synthesis steps, and we map that result back to the Clements taxonomy, we have an actual thing that we can test and study. So that's exactly what we do. I create summary stats looking at what families and genera aren't monophyletic, and we start looking into those. I create some plots so we can just stare at this stuff and try to diagnose issues. And I get them in front of experts like Pam Rasmussen and Marshall Iliff from the Cornell lab. If we find issues, we need to go figure out what's going on. It almost always comes back to taxonomic mismatches between databases. So then we get in there, we fix the issues, and then we stitch all the trees back together and check it all out again. We iterate on this until we're happy, and then we move on to the last two steps. Again, I wanted to take a moment to talk about this standing on the shoulders of giants thing. So critically, OpenTree is tracking all this information, and it's so important for us to track the provenance of all this information for a number of reasons. The foremost one is so that scientists can get credit, the credit that they deserve for their work. So now, for example, we've taken advantage of the information in OpenTree that tracks this to build services on top. So when a user comes along and says, hey, I need a phylogeny for 100 of my study species, it's really easy for us to say, okay, these 3 or these 6 studies were instrumental in providing the information that we relied on to give you that phylogeny, and you should cite them as well. Second, with respect to taxonomic revisions, it means that taxonomists can check which studies do and do not support a given relationship. Someday, I'd really like to be able to link this all the way back to the original museums and specimens and tissues and sequences. that support these relationships. But for now, it's at least easy to move from our final synthetic tree back to the original studies that make those claims. And so if, a number of studies have shown that one genus is actually embedded within another, taxonomists can easily see how well supported that statement is and elevate that to get renamed, that situation to get renamed. It also might mean that somebody sequenced a specimen that was poorly labeled as well. So again, there's reasons to link this to the specimens going forward. And last, the ability to track this information and encourage proper attribution should really help to build a community of contributors. If adding your phylogeny to this collaboration gets you more citations and helps make sure that your hard-won scientific results are improving the science that other people are doing, then this will hopefully incentivize people to participate and build a whole community to keep this project alive going forward. All right, so methodologically, are we there yet? No, I'm sorry. I have a few more steps I need to tell you about. So the tree has 9,233 species in it right now, which means we make precise statements about how around 84% of the world's species are related to one another. This is pretty impressive if you ask me. The Yeats tree, for example, nominally contained all the world's bird species, like I said, but only about 67% of those statements were informed by actual genetic information. Either way, though, we've still got about 1,800 species that aren't in the synthetic phylogeny. So what are we going to do about them? Well, essentially, we use the best information we can find to add them into a reasonable place in the phylogeny. And my starting place here is always birds of the world. In almost all cases, starting from BOW, I could find published information that summarizes what is known about each missing species and offers hypotheses about who its closest relatives are. The related species section is my sort of bread and butter. We can then use this information to identify reasonable positions in the phylogeny where each missing species might belong. These species can be as specific or vague as you like. For example, if the taxon is recently split, barely morphologically differentiated from the related taxon that is in the tree, we're probably just going to bind it in as sister, the missing species in as sister to what it was split off from. But other times we'll only have a guess about what family the taxon is even in. I capture all of this information in a giant spreadsheet, which I'm showing you a brief bit of right here. And I'm going to show you a glimpse of a tricky addition, which is how we add in the two members of the Poliocephalus genus. These two grebes aren't in any phylogeny that I've seen. If you know of one, please point it out. So I had to figure out where to add them. And again, since I didn't have any particular information on this, I made a pretty vague but constrained statement about where to add them. So specifically, what I send off to the addition algorithm says, first of all, add these things into the grebe family and make sure they stay together. That's important. We want the two Poliocephalus species to end up in the same general place in the phylogeny. And then I say, don't break up any of the existing genera like Rolandia, Tachybaptis. And we do this hundreds of times across all the missing taxa to try to capture some of the uncertainty and where these things could end up. And so here, I'm just showing 4 random runs of these sort of addition statements for Poliocephalus. So here they end up as sister to Poticeps plus Rolandia. So Paige and Pam Rasmussen. And then, here they're sister to Tachybaptis. These are all valid positions given the taxon addition statements I gave it. And so we do this for all the missing taxa hundreds of times. And we finally get to the last step in this process, which is now we need to date this tree and match it to the fossil records. So to do that, we match nodes from the published phylogenies to those in the final tree. And we basically smush and stretch branches to average over all those dates from multiple studies. And finally, we end up with a cloud of complete dated phylogenies with one tip for every species in the Clements taxonomy. And a critical piece to point out is that we're creating the synthetic tree in various taxonomies, one for every year since 2021. Also, these taxon addition files are versioned and come out every year as well. And so this means that although the underlying topology is improving, we can revise the addition statements to further improve them. We're always going to end up with phylogenies that perfectly match the eBird taxonomy every year. And all these things are open the inputs are open to access, see, and provide input on. So if you see something, say something. I can fix it. All right. I just covered about 4 years of work in 10 slides or so. So I'm sure some people are snoozing or heads are spinning. So let's take a moment for another fun quiz, starting with the chimney swift. Who do you think its closest relative is?
All right, there you go. It's launched.
Thank you.
I like quizzes. We have to do
Yeah,
more.
they're fun.
One person asked, do the trees contain change info? Was and is, so it was in this clade and now it's in a different one.
No,
Will it retain the historical placement of that species?
I'm not sure I understand the question. The trees that are curated on OpenTree are static and reflect what was published at the time.
Mm-hmm.
We then map those tips to taxa in OpenTree, which is a complication that I'm glossing over. But at any rate, those taxa that we're matching them to can be updated and change over time. So we might call it Picoides villosus when we match it in 2012 or whatever, but now we call it Dryobates villosus. And so those names do update in the database, but the original tree in the database will be whatever the authors published. So honestly, half the time they don't even put useful tip names on the tree. Folks don't, just because it's not expected that that's what they're for, but it's just going to be like a specimen number or an abbreviation. So I guess this is a sideways way of me saying, please make your tip names really useful when you publish your trees.
We hear you loud and clear.
Yeah, but anyhow, long way of answering that question that's not even a real answer.
Okay. All right, here are the results.
Ooh, ooh. People are catching on to my secret tricks.
Yeah.
Fiery topaz is pulling ahead, and that is the right answer. All right. So yeah, sorry, I gotta flash up the results here. I'm frozen. There we go. The answer is fiery topaz. Swifts and hummingbirds are in fact close relatives, part of the same order, the Apodiformes, meaning they have very small feet, both of them. The amazing convergence between swallows and swifts is just something driven by their really similar aerial lifestyles. So impress all your friends with that stat. All right, I think we've probably had enough for the hardcore bioinformatics today, and I'm gonna kind of finish up here. And I'm also gonna stick to fun things for the rest of the talk. So let's think about what we can do with all this cool, aligned, interoperable information. First, the fact that the phylogeny perfectly matches eBird taxonomy means that it's very easy to match both to your life list, assuming you use eBird, and to eBird data at the global scale. So if you want to know if you see a new genus for the first time, a new family, et cetera, we can tell you that easily, but we can also do much more exciting things. So just to get us warmed up to this idea, here what I'm going to show you is a plot of the mean pairwise phylogenetic distance among all the species that have been reported in eBird from each one of these little grid cells around the globe. Excuse me. So I basically divide the world up into a grid. I figure out which species have been reported in eBird from within that grid. And then what I'm plotting here is a measure of the average amount of evolution separating all these species in each grid cell. So blue means that all the co-occurring species in that area are on average distantly related, whereas yellow means that on average all the species in that grid cell are closely related. So the take home here is that regions of the globe that have been more climatically stable over long periods of time tend to have older lineages, and they're composed of sort of more distantly related species. And then as you proceed into more climatically harsh regions, higher elevations, only certain lineages have evolved the necessary adaptations to persist in those environments. So as a result, you can see increasing evolutionary relatedness in arid regions like interior Australia, and then coming up out of the Amazon into the high Andes, in the boreal, going up the Himalayas and into the Tibetan plateau. So that's really neat stuff you can start doing just right out of the gate. But then we can also personalize this stuff. So let's look into where I should go bird watching, me personally, to see evolutionarily distinct species. This is a good way to think about places where you could go to see species, unlike what you've seen before. I had to come up with a few sort of new measures to get there. One that I'm showing here is the average evolutionary uniqueness of all the species in a community as compared with my life list. So this is driven by the average uniqueness of species in the grid cell, independently of the number in each grid cell. It's simply a measure of how unique those species are as compared to my life list. So coastal and pelagic sites really kind of pop here. I'm not much of an ocean dude. If I go bird watching in the ocean, near the ocean, I'm going to see stuff that doesn't look like the majority of my life list. But some other cool stuff pops out here as well. So check out Madagascar, the South Pacific. If I go bird watching in one of these places, I might be seeing slightly fewer birds than species than, say, if I went to the Amazon or the Andes. But most of what I'm seeing is going to be very different than what I've seen before. And so, for example, of the 251 bird families in the world, I'm yet to see 45 of them. 5 of those are endemic to Madagascar. So if I went there, I could see Mesotornithidae, Brachyceracidae, Philippidae, Leptosomidae, Barneridae. I'm sure I butchered a few of those pronunciations, but there are 5 endemic families to Madagascar that I would like to see someday. So this is a really fun way, in my opinion, at least, to link evolution with bird watching and scope out areas where, if I had all the money in the world, I could travel to see unique birds that aren't like what I've seen before. And I'm sure together as a group, we can dream up any number of other fun visualizations and games we might play once these resources are linked into Birds of the World and eBird. So I want to end there and open it up to discussion. Thank you so much.
Awesome. Thank you so much. That's really interesting. I appreciate it. Great. So people are really interested. We've got, and the polls were fun for everybody, I think. So we've got a lot of questions. I'm going to focus mostly on kind of the deeper questions, because we kind of hinted in our discussion that this would be, in our advertising, that this would be focused on more of the technical stuff. So I'm going to start with some of those, maybe not first, but they'll be in here. But I do love to ask about dinosaurs. Somebody did want to know, so let's get that. People want to know how far back on the evolutionary tree that this that the phylogeny goes, and does it go all the way back to dinosaurs? And while you're there, tell me, are birds actually dinosaurs, or are they not? Are they just relative dinosaurs?
Yeah, this is a good question. It's one that I sort of anticipated and looked at a few published phylogenies of dinosaurs and where birds fit in. And I had to admit off the bat that I have trouble remembering all these higher taxon names and keeping it all straight in my head. But yeah, birds descended from theropod dinosaurs, things like velociraptors and raptors, from what I understand. And they have feathers. Those had feathers, so that's a shared feature that unites them with birds. But dinosaur is actually kind of a tricky idea as well. Dinosaur is not really like a monophyletic lineage. In fact, I'm not sure that Dinosauria still exists. So yeah, birds descended from theropod dinosaurs, but dinosaurs itself is kind of a big paraphyletic group of things that went extinct, with the caveat that birds descended from dinosaurs and are very much alive still today.
Yeah, great. So we answered that question. In the past, and I think you might have mentioned it, but we might have missed it. So in the past, there was software to compile genetic sequences and software to analyze them. So what is the latest software to use, and what do you use?
Oh, great question. For somebody else. No, I'm just kidding. I don't know all the great bioinformatics pipelines these days, but there are many of them. And yeah, I'd leave it at that. Our project isn't operating on the genetic sequences directly. And so that we take published trees, which are almost always these days made with genomic and genetic data. Very, very competent people are aligning these sequences and running the models of evolution to generate the phylogenies. But at the scale of all birds, aligning this much genetic information and doing it is pretty challenging. Not to mention, we don't have it for all 11,000 species. So
Yeah.
for the time being, this is the way forward to get there.
Okay, great. Like with Jets et al., will researchers be able to download the phylogenies to, for instance, correct for relatedness easily in analysis? And maybe as a follow-up to that, I wonder if we should show where people can get on Birds of the World. Should we show where the Taxonomy Explorer is? If
Sure.
you click, go there. Do
Yeah.
you want to do that, or should I? You could sort of just show them what we have so far, which is not
Sure.
complete, but
Sick.
I mean, if your vision for how this might go into Birds of the World, I'd love to hear what you think on that.
Yeah.
So someday this will be incorporated into Birds of the World. It might be a year, might be 2 years, but we hope to have it there so that you can, so that you can search species by that. And when you're working on a clade, for example, rather than a species group or a family, it's going to be easier to find that research.
Yeah. So you guys can hopefully see this screen. This is a logged in Birds of the World page. And if you come over here to explore taxonomy, sorry, I've got 2 screens here. You can start scrolling through, all these awesome birds that exist in the world. You can navigate here to family. You can filter, Tinamidae, however you say that, and look at all the tinamus. In some way, it would be amazing, and hopefully we will be integrating the phylogeny into the Explorer page. But if you want to check out really cool phylogenetic visualization tools, check out the OneZoom site that hosts the Yetz tree, actually. And that's really cool. It's a sort of a fractal viewer. So that might be one way that we could do this going forward. There's some pros and cons to a viewer like that, but it's certainly fast and a way to show lots of data. So there was more to that question, but that's the part that you asked about.
So like with Jets et al., will researchers be able to download that?
Yeah. They will. We've got actually in our package, and that basically what it does is you tell it what year taxonomy you want the tree out in. So if you're using AVANET's actually in 2021. That's a big traits database. You might want the tree in 2021. Better yet would be to update AVANET to 2023 and use the tree in 2023. You tell it what taxonomy you want. You tell it what taxa you want, and it's going to pull out a pruned tree for you and tell you what studies contributed to that tree, so you know who you need to cite as well. So yes, but it's not published yet. We got to get that out in the next week or so.
Okay. So can you tell us a little bit how flycatchers evolved? They evolved separately on 2 different continents. So tell me about those. How are they related, or are they not?
Yeah, I mean, they're related in that they're birds, both of them. But New World flycatchers are subausines, part of that lineage that I showed you. So related to mannequins and Phaneroids, all that stuff. Old World flycatchers is a more diffuse group. I don't think they're monophyletic. It depends how you want to define them. But like musicapids are a lot of the old world flycatchers belong to the family Musicapidae. And these things have just converged to look like one another. They're not closely related. Musicapids are not subausines. They're part of later Aucine passerine radiation. And they've just converged to look like one another because they have similar lifestyles, similar diets, aerial insectivores that sort of sally out to catch their prey.
And so perhaps Europeans named them flycatchers because they were similar to what they were seeing in
Exactly. Yeah.
Okay.
Exactly. Yeah.
Great. So can you tell me a little bit more about this concept also with Seriemas and secretary birds?
Oh yeah, they're amazing. Again, it's like a really striking case of convergence in the world. They're both amazing sort of raptorial birds of open habitats. So you've got the secretary bird, which is sort of an iconic species of the savannas in Africa. They're famous for like stomping snakes to death, and they eat lots of like inverts as well. Big prey though, and they've presumably got those long legs. Well, because they do a lot of walking, but also it kind of gets their body up and above the grass so they don't get eaten, and lets them stay away from the stuff that they're
Mm-hmm.
Seriemas are doing the same thing in southern South America in open habitats, but they're super distantly related. Again, totally driven by similar selective pressures to look that way.
Mm-hmm.
Secretary birds are related to eagles and hawks, and seriemas are sort of an early offshoot somewhere around falcons, but I don't think they actually group with falcons. They're just near there in the tree.
Oh,
Yeah.
right. And some people wanted some confirmation on how relatedness is determined. Are we talking simply DNA analysis?
Yeah, usually it's going to be some kind of genetic analysis. I mean, the way these things were done traditionally was you end up with a giant matrix of morphological similarities, and you do things like maximum likelihood, maximum parsimony on these things, and it spits out a tree showing the inferred relationships. Nowadays, we just have giant genetic matrices basically and do similar things on them. Yeah.
Okay. Any thoughts on the dendroica warblers being reassigned to Psittophagus? Yeah. That's
Yeah, it needed to happen. The
Oh, actually, yeah, go ahead. I do have
the
another
actual
question following
taxonomy and the reason why Psittophaga is there, and I know like Niotilta was black and white was somewhere near there, and that was involved in the discussion. I forget the specifics, honestly, but phylogeny mandated some taxonomic rearrangements, and
Okay.
why it ended up as Psittophaga versus Niotilta is probably because a genus name that starts with an MN is not super awesome to name all our iconic birds, but that's just my guess. Yeah.
Okay. I should have asked this one before that. Is the DNA analysis based on Sibley-Ahlquist taxonomy?
Um, um, can you, so
Yeah.
can you say it? Yeah. So like Sibley Ahlquist were some of the pioneers of DNA hybridization work, a few decades ago, and they figured out all sorts of awesome stuff using pretty really creative and also today like rudimentary methods, and figured, I'm pretty sure, for example, the gibber chat, they lent support to the fact that that was probably in a honey eater. So like any number of cool findings like that came out of that. But yeah, nowadays they're actually sequencing the genetics and getting the A's and the T's and all that stuff and lining them up and creating matrices out of it. There it was more of a question of how closely strands annealed and splitting temperatures and stuff.
Okay. In your estimation, how many phylogenists are currently working on this?
Yeah, great question. There's definitely hundreds, maybe thousands of people working on bird genetics. Not that many people know about this project yet, and so I'm pretty excited to launch it and continue launching it, and get some more community buy-in.
Great. Proving yet again, it's just always a group effort, and the more people know, the more information we can get together and just improve what we know scientifically. Okay. One person would like you to explain how you did I don't know if this is too technical, so you can decide, but how you did those map calculations at the end. I'd like to test some stuff myself. So maybe that would be a conversation you would have after, or do you want to just talk about it now?
I can talk about it. It's tricky because of the size of the eBird database, is like not easy to operate on. So there's a pretty fast grid cell thinging device called H3, which is what Uber uses for its maps. And it's not that.
What's it called again?
H3. It's like how Uber divides the world up into grid cells, and it's proven useful for analyses like this. So basically just download the entire eBird data set. And how do I do it? I download the eBird data set, extract single species from that, figure out which grid cells they belong to after having pre-split the Earth into grid cells, and then basically on the fly assemble lots of grid cell lists. So as I pull out a species, I add it to all the grid cells it's in, and then throw away all the raw eBird data. And then I just have a series of matrices of all the cells that each species occurs in. And then I can start doing that. I was actually thinking about making that data available at some point, since it's a lot smaller than the raw eBird data.
Okay.
It's easy to do things once you have that.
Are you going to offer different tree views, maybe including hyperbolic trees?
That's a great question. And I don't know. I would love, to be completely honest, I have never seen a tree viewer that completely meets my expectations. One zoom is my favorite that I've ever found, but I don't like the you can't see tree imbalance in there very well. So like if one clade has 1,000 species and one clade has 1, when you're zoomed out, you can't see that. But I don't know what it's really hard to look at these things and get the detail you want and the perspective you want. And so you need some sort of zoomability, and people are working on this. And I hope that in the next year or two, as this gets incorporated into Birds of the World, we can decide what is best for the community and start implementing some of that stuff.
So my mind goes places. I'm thinking of the Cornell Labs wall of birds that has, representative of every bird in the world. Wouldn't that be great if we had this massive screen, touch screen, that you could just like zoom in and out on an entire wall? It's coming. I tell you what, it's coming.
Yeah.
Let's see. Was hoping that the taxonomy of the Watson would be covered, since it's such an enigma. I'm sure. Yeah, I was going to ask that and I forgot. So thank you.
Yeah, I should have done my homework on where it ends up these days. It's somewhere near Cuckoo's last I checked, but it has its own. The taxonomy is pretty straightforward. Well, depends if it's embedded somewhere. It needs to get revised, but last I heard, it is still part of its own genus, family, and order. Yeah, I know there's people on this call who know the answer to that better than I do, but
feel free to put in the chat if anyone would like to answer that question.
Jake Bird. Yeah.
Okay. What would be a good starting point? Online course, website, video tutorial to learn about how to incorporate bird phylogeny into ecological studies. Would be like, where do you just go for any of that information?
So
you call ELIOT.
Yeah. No, don't. That's not part of my job anymore. There are so Liam Revel has a cool blog that but often
type that name into the chat.
Yeah. Liam and Luke Harmon wrote a book a year or two ago that that is worth a read if that's something you're interested in. That's a great, like if you read that, you'd be an expert. You'd know more about this than I do. So that's a place, but it should start at a pretty general level, I think.
Okay.
Emily Jane's got some information for us. Watson is sister to the clade containing jaconids and what do you call that thing? Limpkin, Aramidae. That's cool. Yeah, they're all weird. All those things are weird. It's the weird clade.
Yeah. All right. Okay. I think that we need one more question, and then well, maybe 2 more questions. I want to say one question for you about biodiversity. Can you talk any more about that new paper from Avian Evolution from Nature, from Stiller et al.?
Yeah, that I'm embarrassed to say I haven't read the paper yet. We do have the tree incorporated into this project, so it is informing the results at the moment, but I haven't actually read the paper yet. So I can't really speak about much more than that, aside from that I know that there's a lot of smart people on the paper and they did a lot of hard work to get to that place, so I suspect the results are great.
Okay. Were swifts and hummingbirds placed under, this is a big name, Capra until recently with nightjars? And what criteria is used to group families together under an order?
It's a great question. What criteria, like taxonomy is arbitrary in terms of like where we make breaks. And I once thought it'd be pretty interesting to write a paper just showing the breadth of like crown ages for genera across the globe. It's pretty broad, right? Like genus is more like a useful idea for lumping species that look together into one taxonomic entity more than it is a measure of how long ago that group of organisms arose. So that's the answer to that question, is that it's pretty arbitrary where we put these. But you know, we tend to put them in sort of more and more morphologically dissimilar entities. I don't think swifts and hummingbirds were ever in Capra mulgiformes. There is a bigger clade, whose name I forget at the moment, that does unite swifts and hummingbirds with nightjars and all that stuff.
Okay. All right, so one more question here. You know, something grave happens every time an entire lineage is lost from the tree of life, you know, in birds particularly. So what are the consequences of this in terms of biodiversity, and perhaps from a more philosophical viewpoint? Like, you know, what does that mean for us when an entire line just ends?
Yeah. So first of all, I would say there's a bit of a ethical conundrum there. I mean, these are things that have been evolving on their own, doing their own, having their own evolutionary history and exploring the earth for millions of years. So, you know, I'd say there's, and when any species goes extinct, it's pretty tragic. When entire lineages that have sort of like been off doing their thing for millions of years go extinct, there's a bit of a sense of even more profound loss there, right? But then in terms of the actual functional relevance of that, different lineage, like diverged lineages tend to have different traits, tend to perform different services in the ecosystem. And so, you know, the more of these distant lineages we lose, the more we're losing novel functional traits in the ecosystem, presumably. I get a little leery when we start trying to put value, like actual financial dollars, on species myself, but that is an argument one can make, and up to a point it's true. We're losing, like if you lost all the flamingos, you would not have any birds that filter fed with their heads upside down. That would be gone. But I would be more sad that that group went extinct, period.
Yeah. Okay, well, thank you so much for coming today. I really appreciate you being here, and everybody else who came and watched. If we didn't get to your questions, I'm so sorry, but you will receive our, you know, the video to this next week. And we might put in a few of those resources, the links in it as well. Let's see, Jess, do we have anything else? As always, we put out a feedback form if there's anything you'd like to share or tell us about what you're doing or how this relates to what you're doing. That's always wonderful to hear. And contact us at support@birdsoftheworld.org if you have any questions to share, or if you want to give a webinar to our Birds of the World audience next year. So with that, I think that ends it for the day and for the season, sadly. So we'll see you next September as we start again with a 3-part eBird workshop. Thank you. Have a great day. All right. Thanks again, Elliot. Thanks, Jess.
Thank you all. Thank you all. Bye-bye.